All right. So let's talk about HIV, human immunodeficiency virus. So today is HIV lecture. So what is HIV? HIV is a single-stranded RNA virus. So usually RNA is a single strand, and HIV basically a single-stranded RNA virus. One thing notable about single-stranded RNA, this HIV virus, it is a retrovirus. So it is a family member of retroviruses and subfamily of lentiviruses. So how does HIV look like? What is the structure? So basically there are three main structure of HIV. The first being envelope proteins. So the envelope proteins, keep in mind that GP120 and GP41, they are really critical proteins. GP120 here is the surface envelope glycoprotein. Let me get the pointer here. GP120 here is the surface glycoprotein 120, and then there's GP41 is a transmembrane envelope glycoprotein. They are really crucial when we talk about the HIV replication and HIV life cycle. The crucial proteins on the cell surface, on the envelope, and they help attach the host cell receptor. And so CD4 receptor T cell is an example of a receptor that these proteins attach, that GP41 and 120 attach to. And then inside the envelope is another layer, capsid. It's called capsid. So you can see here capsid protein P24 and P17. Capsid protein P17 is here. Is one of the matrix protein or capsid in the capsid layer. And then inside, if you go further inside, is the core of the virus. The core of the virus has capsid protein P24. And then once you even go further inside, there's HIV genome, single-stranded RNA. So basically, this is a single-stranded RNA, and there are additional proteins, additional machineries in the virus, like reverse transcriptase. Remember, the HIV is an RNA virus, so it requires this reverse transcriptase enzyme to convert the RNA to DNA once the virus replicates and gets inside the cell, whole cells, and then start replicating and integrating its viral RNA into the host nucleic acid. So reverse transcriptase, and then there are proteases, USC protease. As the name implies, it breaks down proteins. So we'll get into that in a little bit. And then integrase protein is basically it helps integrate viral nucleic acid with the host nucleic acid once the virus infects the whole cell, the whole cell. And what else? So all these essential proteins are here, but the three main structure is envelope, capsid, and core, and then the genome. And of course, all these proteins are encoded by three genes, three major genes. So the first gene being envelope gene, ENV. So envelope gene encodes GP120 and GP41. And then another gene called GAG, group associated antigen gene. GAG, it encodes the core proteins GP17. That's what we talk about. P24 and P17, and then P9. P9 somewhere here is one of the proteins inside the virus. And another gene, the third gene, is polymerase gene. So as the name implies, polymerase means that it helps It helps with the viral nucleic acid amplification, nucleic acid, building up the internal nucleic acid machinery. So, and the polymerase gene will encode protease, endonucleases, integrase, so any kind of like nucleic acid amplification and nucleic acid life cycle activities. So it will encode. They are encoded by Paul. Then the reverse transcriptase is a bifunctional enzyme. So as we talked about HIV, as I alluded to earlier, HIV replication and pathogenesis. So how does HIV HIV virus? How does a virus get into the host cell? So remember all those proteins that we talked about. The envelope protein GP120, it plays an essential role in fusing with the host surface protein. For example, here the host cell that we're looking at is CD4 T cells. CD4 T cells. So it has the receptor CD4 on the surface. So this is host receptor CD4, and GP120 attaches the CD4. And then, with the help of some other co-receptor on the host cell, one of the co-receptors is CXCR4 or CCR5. So, with the help of that, and the virus attaches to the host cell, and then it fuses with the host cell membrane. And this is where the protease enzyme comes into play. So once the viral membrane fuses with the host cell membrane, it breaks down there has to be a breakdown of this fusion the fusion between the two cells. So proteases will break down this little junction, so that way the viral nucleic acid machinery will get into the host cell. And once it gets into the host cell, what does it do? The virus is gonna replicate, right? So in order to replicate, it has to replicate their nucleic acid. So that is the essential component. So speaking of nucleic acid, the central dogma is that from DNA is a double-stranded nucleic acid, deoxyribonucleic acid. So this is a central dogma that the cycle of the nucleic acid amplification in eukaryotes and in prokaryotes, it starts with DNA, and DNA transcription happens. So the word transcription from DNA to RNA, and then RNA. And from RNA to protein is translation. So messenger RNA, tRNA, transport RNA. So with the help of any of those of RNAs, the RNA is translated into proteins. Protein amino acid then builds the protein building blocks. So this is the central dogma. So here in HIV replication, once the viral RNA gets into the host cell, RNA is a single-stranded RNA. But in order to keep infect the host cell, it has to integrate with the host DNA. The host, we do have DNA, right? So in order to integrate it, so this RNA, viral RNA, has to be converted to DNA. So this is where reverse transcriptase can come into play. Reverse transcriptase will convert the RNA to DNA. So viral DNA is formed by reverse transcription. And then once the viral DNA is formed, then it goes into the host nucleus, and it integrates with the viral DNA. It integrates with the host DNA. So this is where the integrase comes into play. So it helps integrate the host DNA, and so it becomes double-stranded DNA. So one thing strange about HIV nucleic acid assembling machinery is that it has ribonuclease H activity. So what does it mean? So basically, any living cells should have double-stranded DNA or two strands of DNA, or either two strands of DNA or something like viruses, like single-stranded viruses like HIV or influenza or any viruses. It will have single-stranded nucleic acid versus double-stranded DNA. But if there is RNA and DNA, like this, it became three strands. If there are three strands here inside the viral infected host cell, then ribonuclease H will degrade RNA, which means this RNA is an extra piece inside the host cell in order to continue infecting the some other host cells. So basically, once the DNA is formed, once the viral DNA is integrated with the host DNA, and then the new viral RNAs are formed, and then it follows the central dogma again, from DNA to RNA. And then these RNA then becomes like all the viral proteins, because you have to assemble the viral proteins, right? Because now the viral nucleic acid is amplified. So the viral protein has to be assembled, and then it basically then packaged. It got packaged as a virus. With the viral protein, what is it? The envelope protein, capsid, any of those viral proteins that we talked about. So it has to become a complete virus. So the virus, with the help of human genome, and then its component, it makes additional proteins. And then a new viral RNA and proteins, they move into the cell surface of the host cell, and then they form a little vesicle, and then that's where HIV forms. And then any of those surface proteins are formed, and then it goes into the surface, and then it got detached from the host cell, and it became a mature virion. Then the virion will keep on going into the bloodstream, and then it keeps infecting more cells, and then this cycle keeps this cycle keeps ongoing. This replication cycle just keeps on going like this. So it is interesting that if you remember this step by step, like what proteins are involved, what each step, and this is really essential, crucial. If you remember this, and then you don't have to worry about the treatment, because the treatment is based on the life cycle or the replication cycle of HIV. So this is the life cycle, and it's all replication or pathogenesis of HIV. And what about any additional regulatory genes that HIV has? It has 6 additional genes that regulate viral expression. These are important in disease pathogenesis. One being TET, transactivation or transcription gene, REV, regulator of expression of virion protein, VIV, viral infectivity factor, VPU, VPX, NEV, VPR. So you just keep reading it on your own. So these are additional regulatory genes. So we talked about the viral infection, like how does HIV infect a host cell. So what is going on at the very cellular level? Who really sees the virus in our body? So the first cell, in our immune system, that sees the virus is actually antigen presenting cells like monocytes or macrophages or dendritic cells. So these guys are the first line because these are the innate immune cells. They see the virus first. They will process it. They take it in here. They take it into their intracellular. So this is one of the antigen presenting cell. For example, macrophage. Macrophage is all those like dendrites, sticking out from the body. women's cell body. So dendritic cell is another type of APC, and antigen presenting cell. So it takes up the virus and it processes the virus into like tiny little pieces. So basically, it breaks it down into tiny little pieces to present antigen, to present these proteins to T cells. So T cells, remember, T cells are lymphocytes, right? CD4 T cells, CD8 T cells, they are lymphocytes. So antigen presenting cells, as the name implies, they present the antigen to T cells. Depending on the major histocomplex protein class 1 or 2, MHC class 2 is presented to CD4 T cells versus MHC class 1 to CD8 T cells. So these are all different receptors involved in this. So what is going on at the cellular level in the macrophages or monocytes is that they, just like the virus infect the CD4 T cell, the viral protein GP120, GP41 envelope protein, it binds with the host cell receptor, macrophages or monocyte cells receptor CCR5 chemokine, those, and then the co-receptor CCR5 chemokine receptor. And one thing is that monocyte is refractory to the cytopathic effects of HIV, which means refractory basically means is resistant to the cytopathic effects. So HIV pretty much HIV loves CD4 T cells. That's why, anyone with HIV, they have really severe, chronic HIV infection has CD4 T cells count really low, versus they don't really have impact on monocytes. So monocytes is refractory to the cytopathic effects of HIV. And one thing is that virus can hide in these monocytes, and they can survive in these cells, and then can also be transported to various organs in the body. And one other reason is because monocytes and macrophages, they are the first line, the first defense of the immune system, innate immune cells. So they being able to survive in these cells, that helps the virus to just make it, being able to disseminate to a lot of all different parts of the body. So effective immune surveillance does not really develop since HIV hides inside them. Macrophages, they serve as a major reservoir for HIV in the body, just like I mentioned. And important consequences for the development of neuropsychiatry manifestations and interstitial pneumonitis by infection of pulmonary alveolar macrophages. So macrophages, monocytes, they are pretty much everywhere, like tissue macrophages, like in our lungs. There are lungs alveolar macrophages, right? So they can just hide there, and then pneumonitis can develop. So that is what is going on at the monocytes or macrophages intracellular level. And then in the lymphocytes, we already talked about it. So T cells are really the cardinal cells that HIV likes to infect. So the cardinal feature of HIV infection is that once it infects, and then it just destroys it. So it's depletion of T helper lymphocytes. And the major receptor I talked about is CD4 for CD4 T cells, and then co-receptor CXCR4, and the receptors on lymphocyte that binds with the GP120. So we talked about it in the second slide of the of this talk of this lecture, and in the HIV replication, HIV life cycle. And so the death of HIV infected cells is also because of the immunologic attack. Why? Because CD4 are helper T cells. On the other hand, CD8 T cells, they are cytotoxic T cells. So as the name implies, cytotoxic, they pretty much blows up the any T cells or any T cells with infected organisms. For example, here HIV. So cytotoxic CD8 lymphocytes and the induction of apoptosis mechanism. Just by the induction of apoptosis, it just kills it kills the cells. So that's what happened when HIV infected cells is HIV infected cells dies from the apoptosis, dies from the our own immunologic response. And then CD4 helper T cells plays a critical role in the immune response, as we already know. And so what are the clinical manifestations? So the very beginning of HIV infection is manifested by the acute retroviral syndrome. That is a very initial manifestation. So acute retroviral syndrome basically starts off with non-specific symptoms like mono-like illness. So mononucleosis, mono-like illness like fever, joint pain, adenopathy, pharyngitis, rashes. So any of these like mono-like illness, this is the first initial manifestation. Sometimes symptoms resolve within 15 days, depending on individual immune system, immune response, how strong someone's immune response is, or if someone has already an underlying impaired immune response, their symptoms might not resolve within 15 days. It might progress to severe or full-blown HIV infection in a very short amount of time. So these are basically the symptoms or signs, and then the HIV primary infection happens. So primary infection basically means that someone is really sick now. The virus, it goes into the cell. Remember, it starts replicating in the cell. It starts going into once it multiplies. Once it multiplies with the nucleic acid and then making making the viral protein assemblies, and it makes lots and lots of HIV, and then it infects more and more cells. And once it starts multiplying more and more, the virus in the body, the viral level becomes higher and higher, and then the body starts showing up as viremia. Viremia basically is the virus in the blood, right? And someone is sick with sepsis with the virus. So viremia is detectable for about 8 to 12 weeks of initial infection. And then lymphoid organs will also get seeded with viruses by that time, because the virus replication is really efficient. And so it goes into the lymphoid organs. And then with the amplification, these two really go hand in hand. Once the viral amplification takes off, and then the CD4 T cells number would drop. So these two go hand in hand. Significant drop in the CD4 T cells, and immune response occur. Plasma viremia drops, and then the levels of CD4 cells. If the immune response happens, the plasma viremia would drop, and then the level of CD4 will rebound. That depends on someone's immune response, just like I mentioned. And the immune response is, if the immune response is not really, someone's immune response is not good, is not unable to clear the infection in the initial phase. So, and then the HIV infected cells just persist in the lymph nodes, that they just keep multiplying, and they just keep going into some other parts of the body, some other parts of the lymphoid organs. So this period here, if the virus persists in the lymph nodes or any lymphoid organs, the period of clinical latency can happen. So it may last for as long as 10 years. So during this time, there's a high level of ongoing viral replication. So even though it's latent, it will keep replicating, but it will just stay, it's kind of like stay low, lay low. So the patient would develop constitutional symptoms. So constitutional symptoms, anyone knows what constitutional symptoms is? It's basically constitutional symptoms is really non-specific symptoms like fever, lethargic, headache, viremia, any of those like non-specific like symptoms, like constitutional symptoms, and those are really clinically apparent. And as it keeps on going, as the infection keeps as the infection progresses, the patient will have a generalized lymphadenopathy, and it goes into the oral. The clinical picture became more apparent, like oral diseases like thrush, like Candida thrush is really common, and then periodontitis, gingivitis, oral hairy leukoplakia. And then also on the skin, there's some dermatology consequences. The skin started because it goes everywhere. So it's not just the virus going everywhere. Because the immune also drops, right? So CD4 drops, the immune system drops. So there's chances of a cutaneous infections like HSV, VCV, molluscum contagious, and scabies. So any kind of cutaneous infection happens. So these are dermatologic consequences. And then renal, and when the virus also goes and replicates in the renal tubular cells, then the HIV-related nephropathy, proteinuria, and then the serum creatinine will increase, and then there will be glomerular injury, and eventually tubular degeneration. So all these all these happen in the advanced HIV status. And then cardiac too, it also attacks the heart, accelerated atherosclerosis, MI, myocarditis, pericarditis, and then all those cardiac markers will also increase. So one of the clinical manifestations is really important, really important, is iris. So iris means immune reconstitution inflammatory syndrome, iris. So what does iris mean? Iris is basically, again, non-specific constitutional symptoms like fever, night sweat, jaundice, fatigue. So any of these non-specific constitutional symptoms in HIV patients who have at least for about 8 weeks of antiviral therapy. So once the patient gets on the viral treatment, and once they have about 2 months of antiviral treatment, and that's when they a fair number of those patients would develop iris. And along with the iris, as the name implies, again, so anything you just like try to decipher what the meaning is and what the clinical manifestations are based on the name. So immune reconstitution inflammatory syndrome. So immune, so because of our own immune response against the virus, and with the help of the antivirus, because antivirus also helps suppress, also helps kill the virus. At the same time, our immune system also start rebounding, and along that line inflammatory responses happen, and along the line opportunistic infections also happen in iris. TB, MAC, Mycobacterium avium complex, CMV, Hep C, and cryptococcal neoformans, cryptococcal infection. So these will happen. So we will cover those later part of the lecture. So what is the epidemiology of HIV? So HIV, we know HIV is it happens worldwide, but there are typically two genotypes, or two main types, not just genotype, two main types of HIV. HIV-1 and HIV-2. HIV-1 is pretty much found in worldwide. In our country, HIV-1 is pretty much is HIV-1. HIV-2 is very, very specific and unique to only Western Africa. And then once you can again classify further in further HIV-1 into a further classification. So HIV-1 has again subclasses like M, O, and N. So these are like subclasses based on their like different groups, like genome components. So and then M and O, and then N is non-M and non-O. And then there are also other subgroups like non-M and then group P. And M group have also 11 subtypes. And then you just keep subclassifying the HIV-1. So basically they are all HIV-1. So you just keep subclassifying. And then HIV-2 has 8 subtypes, A through H. So again, HIV-2 is more common, is mostly unique to Western Africa. But then with the worldwide, worldwide increased travel between countries, among all different countries, HIV-2 is, the people with HIV-2 can also travel from Western Africa to other parts of the world, and you can also diagnose HIV-2. In Thailand, so in Asia, basically subtype B is actually HIV-1. It's mainly through IV drug use, and the subtype E is mainly through sexual transmission. So those are just notable points that are shown in the literature. And then HIV-1 group O is originated from Africa, and then they are different from group N strains. So why do we care about all these HIV subtypes? And one important thing is that when we establish HIV diagnosis by lab serology and lab tests, sometimes it can be missed depending on, say, HIV, depending on how a lab test is developed, or how depending on the manufacturer when they make a test, what protein type they are targeting. So HIV, basically they target HIV-1 because HIV-1 is found worldwide. But the fact that someone with a really severe HIV clinical manifestation, because even with the HIV clinical manifestation of HIV, it's not really specific. It's not really specific to HIV because it's very like constitutional symptoms. Remember. So it can be missed if someone from, say, someone from Western Africa with HIV-2 comes and present with with all these non-constitutional constitutional symptoms. So it can be missed. So this is why just more of a background, additional information for you to understand their subtypes of HIV-1. Different types of classes of HIV-1 and 2, and then subtypes of HIV-1, subgroups of HIV-1. So depending on the serological assay and depending on the type, it tends to produce indeterminate, like Western blot patterns or confirmatory testing. So we'll talk about the lab tests in a little bit. And so we mentioned the HIV diagnosis, right? Because HIV, just by clinical manifestation, is so non-specific. You have to have the diagnosis by the laboratory test. So in order to do the diagnosis of the laboratory test, we have to know when in our body what markers we start seeing them. So time 0 here, HIV infection, time 0, and it takes about over a week for the viral RNA to show up in the blood. So so once it starts showing up, it's not even it starts going low, and then it will start it starts shooting up. That's when like the virus is actively replicating. So this is HIV-1 not just one HIV RNA detectable in the blood. Viral detection, it can be done by nucleic acid tests, because as the name implies, the RNA, nucleic acid, is detectable at this time point. You do it by nucleic acid amplification test, nucleic acid test, and so because the virus starts replicating and amplifying really robustly, then we start seeing the P24, the core, and then the P24 antigen, the core antigen. So we start seeing it in the blood. So that's also about like 2 weeks. So it takes time. There's some latency here. There's some latency that the window can be missed. There's a window, the HIV diagnosis can be, diagnosis can be missed. And then what about antibody? So it takes time for our immune system to process and to make antibodies. So it takes about 3 weeks, like a little over 2 weeks. Actually, almost 3 weeks. Between 20 and 30 days. That's when HIV antibodies start showing up in the body, in the blood. So antibody detection happens here. So like as here I highlighted this time period here. There is a seroconversion window. Seroconversion means that when you start seeing the antibody in the body, in the blood. So seroconversion window is such a big window here. If you're just relying on the HIV antibody to establish the diagnosis, you will miss it here. Like the first 21 days or the first 3 weeks, not just 21 days, the first about 3 weeks. So here I wrote down, I have it here, 3rd generation, 2nd generation, 1st generation. What does it all mean? So the higher the generation, these are all immunoassay. 4th generation immunoassay, 3rd generation immunoassay. The higher the generation, the test platform generation. So this is when you detect these 4th generation, 3rd generation. These are all antibody serology detection assay, immunoassay, serology assay. So the 4th generation is the highest, and actually 5th generation is the highest out in the western part of the world. So the higher the generation, the earlier that you can detect the antibody, and also depending on how the assay is designed, you can also detect the antigen as early as possible, the higher the generation it is. So we'll talk about it in a little bit about the immunoassay HIV test evolution, like how it evolves. The 1st generation immunoassay is the very, very, very first, like the ancient assay that was out in the market a long time ago in the 90s, or late 90s, is that it cannot detect the antibody in the blood until like day 50, which is about 2 months. So see how risky it is in like giving someone with a possibly false negative results. So with the technology, the HIV detection, the immunoassay generation, the test generation have evolved. So here is an eclipse period, and then the acute HIV infection happens in this period here. Why? Because the virus actively replicates. Then that's when we see viral RNA, protein, all in our blood. So this is when the virus is like actively replicating. That means acute HIV infection is going on, and then that person has the established HIV infection. Once it has established, viruses in our body, just established replication and ongoing cycle in our body, so establish HIV infection throughout. So what about the test? So OraQuick, there's one rapid test, OraQuick Advanced Rapid Test. It is a lateral flow assay. I think that in the general hospital they use lateral flow assay. This similar, or if it's not the same. It detects both HIV-1 and HIV-2 antibodies. So it depends on the manufacturer as well. So typically it detects HIV-1 antibodies, but it does not differentiate between HIV-1 and 2. So it is based on the enzyme-linked immunoassay. So the sample, you put in the sample here, it has the HIV-1 and HIV-2 specific antigen coded on in the device, in the machine, not the machine, in the device. Then what it detects is if you have HIV antibody in your blood, or oral fluid, and the antibody will bind to the antigen by enzyme-linked immunoassay method. So it uses synthetic peptides derived from the HIV envelope region and GOAT anti-human IgG. So this is the technology, how the test is designed. And then what kind of specimen type you can use? Oral fluid, finger stick Western blot, finger stick whole blood, venipuncture whole blood. And the result is really quick, 20 minutes. What about the sensitivity, specificity? So when you think about a test to establish a diagnosis, you always want to think about sensitivity and specificity. How sensitive is the test? How specific is the test? So it depends. So this is actually third oral quick advanced rapid test is a third generation immunoassay. So if you remember this graph, third generation immunoassay, they are fairly good. They can detect the antibody pretty much way a little early of the infection period. So this is rapid test. And how do we really establish HIV test?